use is these valcyclic phosphate molecules that tend to be chain terminators. In other words, as these get incorporated into the DNA chain, it causes DNA to stop replicating and it causes cell death, necrosis, and apoptosis. And the patient then goes on to have temozolomide. So again, this cartoon just shows the scheme again. At the time of surgery, we take out the tumor, then we inject this adenoviral freehand injection, this adenoviral vector that expresses thymidine kinase. The patient then takes for 2 weeks valacyclovir, the drug. This gets converted into a nucleotide analog. Associated with radiation, this basically gets incorporated into the DNA chain, causes cells to die. In the process of death, tumor antigens get released and recognized better by the immune system, and you get this immune system cell jumpstart with activation of T cells that will come in and scour the tumor to try to get long-lasting immunity. And we call it immune response and surveillance. So this is a clinical trial that went on for quite some time. In a phase 1 trial, we actually showed that it was safe. Phase 1 trials are usually done to establish safety. And then we went on to a phase 2 trial that was multi-institutional, involved multiple institutions in the United States. And in this trial, we tried to show whether there was any evidence in a small number of patients. Was there any signal, evidence of efficacy? And so in this group, again, patients got GMCI plus standard of care resection. And to show whether there was any efficacy, we actually enrolled in a prospective fashion a group of matched controls that did not get GMCI but just got standard of care. And these were enrolled at the same time, prospectively and historically, at another institution. And at the end, we had 48 patients that completed this combined approach, and 134 patients that were matched to these patients that only had standard of care. And this was a way to try to get a sense whether there was any efficacy from this trial. I'm going to give you the punchline because this has been published. It was published in Neurooncology initially in 2016. This is when the paper first came out. I think the final paper came out in 2017. But again, it was a phase 2 multi-center study, and Ohio State, City of Hope, Methodist Hospital, and as well as University of Chicago and the Brigham participated in this trial. And so these are the survival curves. And the punchline is the one we went back and looked at the survival curves. There was one group of patients that seemed to be doing best. And these are the patients that had a gross total resection of tumor. In other words, if after surgery, you could show that most of the enhancement, in this paper, we said 95% of the gadolinium enhancer had to be removed. These are the patients that did best. So I want you to compare the red curve. There were 18 patients in this red curve that had gross total resection, but also had GMCI plus standard of care. And the median survival time of these patients was 25.1 months. And when we compare it to patients that only had the gross total resection, but only standard of care without GMCI, there were 44 patients in this group. Their median survival time was 16.3 months, and this is the blue curve. And these were statistically significant differences. If you were a patient that still had gene-mediated cytotoxic immunotherapy plus standard of care, but only had subtotal resection, in other words, there was gross disease left behind, there was no evidence that GMCI was better than just standard of care alone. And these are the dashed curves showing that there was no difference in these two groups. So again, this was data showing that there was a statistically significant improvement in survival if you had a gross total resection and GMCI compared to a gross total resection with just standard of care. And again, it's just a phase 2, so it's not a phase 3. These were not randomized patients. And so before we can be more sure about this data, we will need to do a large phase 3 trial, which is currently finally in the works. We're planning for it to show that there's proof and that there's evidence that these survival data are maintained. I think even more telling, instead of just looking at Kaplan-Meier survival curves, is to look at what are called landmark survivals, where we look at survival of patients at 12 months or 24 months or 36 months. So here we're looking only at the GBM only group and the patient that only had a grossal resection. And what you can see here is that if you had GMCI plus standard of care, there were 18 patients in this group. At 12 months, about 89% were still alive. Compared to standard of care, only 63.6% were still alive. If you looked at 2 years, over half were still alive versus 27% with standard of care alone. And at 3 years, over a third of these patients were still alive with less than 5% in this group alone. So again, this is data showing that there seems to be some signal, and I call it a signal, that there's efficacy. So even though there seems to be some value in doing GMCI alone, we still have a lot of patients that die. So can we make this better? So at the time that we were doing this trial, there was a lot of interest in immune checkpoint inhibition. Immune checkpoint inhibition is a way to stop T cells from getting exhausted. There's this entire concept that when you try to activate T cells against tumors, the chronic exposure to tumor antigens makes T cells anergic and exhausted. It's also thought to be one of the reasons why GBM can be so immunosuppressed, because the T cells just don't work well enough. So we became interested before doing the phase 3 trial to see if we could combine GMCI with immune checkpoint inhibition. So we went back to the lab, and I'm not going to give you any mouse experiments, but we went back to the lab and actually showed that you could combine this gene-mediated cytotoxic immunotherapy in mouse models with immune checkpoint blockade and show that the two together were better than either one singly. And so based on this, we then started another clinical trial where we combined nivolumab, which is a PD-1 inhibitor, to the same GMCI plus standard of care treatment. Now, again, this is a newly diagnosed glioblastoma patients, and they get surgery plus GMCI plus chemoradiation, but they also have nivolumab being provided. This is still a phase 1. We're just trying to show that the two things together are not too toxic. And I'm not going to go through all this, but this trial is supposed to enroll 36 patients, and it was started in a multi-institutional fashion about a year and a half ago. And this just shows you all you have to do to get stuff through clinical trials in the United States. Maybe it is easier in Brazil or in Latin America. But, we started writing the initial clinical trial protocol in 2015. We actually wanted to get nivolumab, the drug, for free, and Bristol Myers Squibb decided to allow us to gave it to us for free, basically to give it to patients. Because this is a trial that was being supported by the NIH, we had to submit a protocol to this consortium called the ABTC. We then had to resubmit this again. We also had to submit it to the NCI. Again, there were several rounds of this, and they finally approved the protocol. And we had to go through all these other ways. Unfortunately, right after 2 years of doing this, Bristol Myers Squibb decided that they were not interested in glioblastoma anymore because the phase 3 trial of nivolumab alone had failed. And so we had to go back and try to get this drug for free again, and they finally decided yes. So we were finally able to accrue the first patient here in spring 2018. And before COVID, there have been 21 patients out of 36 accrued. So this trial is still ongoing. The 4 main accruing sites were Henry Ford, Penn, the Brigham and Wake Forest. There's a couple more that have accrued now. But now even though clinical trials are still allowed to continue, we're clearly going in a much slower fashion. And my understanding is there have been a few more patients accrued, but we're still not up to the 36 patients that we need to do. So I hope the next year you'll bring me back so I can tell you the story of this trial, or whether the two things together work better than either one alone. So my first conclusion is that GMCI, this gene-mediated cytotoxic immunotherapy, we use this adenoviral vector to deliver thymidine kinase, shows encouraging overall survival in this multi-institutional phase 2 trial in newly diagnosed glioblastoma. And again, there's very, very few trials that happen in newly diagnosed glioblastoma. And so I think this is fairly unique. When we see failures and patients fail, this may be to the point that immune checkpoint signaling gets upregulated. So again, before going to the phase 3 trial that we're planning, we're now currently pursuing the combination of this plus immune checkpoint blockade. And we're accruing patients to this. So I told you about this part, but as you know, most clinical trials happen at recurrence. Now, the clinical trial arena has always been the purview of the medical oncologist or the neuro-oncologist, the radiation therapist. And I think one of the lessons, or one of the things I would like to tell the neurosurgeons is we have to be involved too, not just to procure tissue, but to actually do the trial ourselves. And again, injecting things into the brain is what we do best. And so this is a way for us to not only get patients, because patients come and see you because they have really no other option, and so they want to get their tumor out, but also to try to learn something from patients by doing clinical trials. So the second clinical trial I'm going to tell you about is this one right here. And this is another adenoviral vector that makes a gene called interleukin-12. And this is a trial that we finished and we actually published already. And so in this trial, what we want to do is deliver the gene for interleukin-12. And why do we want to do that? Turns out interleukin-12 is a very powerful cytokine. It's an amazing immunoactivator. It's been actually called the master regulator of the immune system. When it is made primarily by activated antigen-presenting cells, this in the brain could be anything. It could be tumor cells. It could be astrocytes. It could be microglia. Interleukin-12 immediately turns T cells into activated T cells that make lots of interferon gamma, makes NK cells, make lots of interferon gamma. It reprograms myeloid-derived suppressor cells into a M1 phenotype. It also may have some anti-angiogenic effects. And it makes B cells make more antibodies. So it really does a number of different things. When interleukin-12 was first discovered in the late 1980s, clinical trials of using this as a peptide, in other words, delivering this systemically in patients with metastatic cancer, were met though by quite a lot of toxicity. Patients develop what's called a cytokine storm syndrome or cytokine release storm syndrome, where basically there was just too much interferon gamma made, as well as other cytokines. And the toxicity was at the level of the liver toxicity, kidney toxicity, cardiac toxicity, and therefore clinical trials of using interleukin-12 alone were stopped. So in order to get around that, two things were done. Number one, we wanted to deliver the gene inside the tumor. So instead of getting interleukin-12 systemically, do it by using a gene therapy vector or viral vector like adenovirus. So the virus would actually be injected into the tumor, and then tumor cells that were infected would make interleukin-12. So that potentially could reduce the systemic toxicity. But the second thing too is we wanted to control it. And the way we control this is by actually turning off interleukin-12 when it infected a cell, by using what's called a switchable or regulatable promoter. The promoters are parts of the DNA that actually turn on the expression of a gene. So we had a promoter in this vector that basically turned off interleukin-12, and it would only turn on if the patient ingested a drug. The drug is called Veldimex, VDX, that actually went and turned on expression of the gene. And this is explained a little bit better in this cartoon, where you have somebody with a nice British accent, more than an Italian accent, telling you. So
with IL-12 expression, the tumor is injected with AD-RTS-HIL-12, a genetically engineered, non-replicating adenoviral vector. The virus implants genetic code for IL-12 and the Rio switch to regulate IL-12 expression. In order to turn on local expression of IL-12, the patient takes a capsule called Veledimex. When patients take Veledimex, expression of human IL-12 is turned on at the tumor site. When they stop taking Veledimex, IL-12 production stops. The amount of Veledimex controls the amount of IL-12 expressed. IL-12 recruits an immune response, such as T cells, directly to the tumor environment to destroy tumor cells.
So that's a cartoon that kind of explains.
IL-12.
And so again, just a few more slides of explanation. So again, we want to, at the time of surgery, we're recurrent GBM patients, the clinical trial was to resect the tumor and then inject the cavity with this adenoviral vector. It's got this fancy name, AD-RTS-HIL-12. The important thing is the IL-12. But IL-12 is actually not being made because RTS is this switchable promoter. The company is called Inmunotepria, and it's called the Rio Switch Therapeutic System. And this actually does not make sure the IL-12 does not get made. The patient has to take this drug by mouth, which goes into the across the blood-brain barrier, binds to this promoter and turns it on. So now you do make IL-12. And the IL-12 gets made over a period of about 12 hours. And that is like a beacon or an alarm system that makes T cells come and scour the tumor to try to get rid of the tumor cell. So this is what mouse experiments showed to be or showed to happen. We actually had to prove that this was true in humans. So this was a phase 1 study of IL-12 vector-mediated gene therapy in recurrent progressive glioblastoma. These were the 5 participating sites: the Brigham, Cedars-Sinai, University of Chicago, San Francisco, and Northwestern. And again, these were patients that had a recurrent GBM. We took patients at first, second, third recurrence. We even took patients that had multifocal disease. So there was a lot of patients with a lot of different tumors types that would come in, all GBM. And one of the first questions we wanted to know was in mice, we know that Veledimex, the drug, did cross into the tumor, but we did not know if that was true in humans. So before surgery, the patient took 1 dose of this drug, usually about 3 hours before the scheduled resection. And at the time of surgery, after we resected the tumor, we injected the craniotomy with this vector. And then the patient from day 1 to day 14 took the drug to turn on expression of interleukin-12. Now, this is a phase 1. We're trying to see if it's safe. And we also were trying to find the right dose of the drug to give. So we actually started at a dose of 20 milligrams of the drug. This seemed to be well tolerated. So then we skipped and went up to 40 milligrams. Turns out the 40 milligrams was way too toxic. There was actually too much interleukin-12 getting made. And these patients did get what's called a cytokine release syndrome because interleukin-12 was spilling over into the blood and causing liver toxicity and other problems. So we actually went down to 30 milligrams, and this was also too high of a dose. And so we went back down to 20 milligrams, and this was the final dose that we decided to go with. In some patients, we also did 10 milligrams to see if that was better than 20 milligrams, but it actually was not. So the dose is 20 milligrams of Veledimex And there was a group that got 40, 30, and 10, but they really could not finish all of the drug because of toxicity. If you're interested, this paper was published last year in Science Translational Medicine. These are all the co-authors. I also must tell you that this is the very first time in humans that we actually could show that you can regulate through a drug a gene. And this really makes you think that there are a lot of other applications, not just in this particular paradigm, but you can see a lot of things where you want to express a gene in a tissue, but not always. And you may want to take this drug to express this gene in a tissue just based on taking the drug itself. So this is sort of some of the key data to show that what we think we're doing, we're actually doing. So this summarizes this the key data. So again, when you take the drug, when you do not have the drug, you're off. If you take the drug, you're on. And here we're comparing patients that took 20 milligrams, which was the dose that seemed to be safe, with 30 milligrams and 40 milligrams. So the first question, we take 20 milligrams, 30 milligrams, and 40 milligrams, you see more drug in your plasma? Yes, you do. So there's a dose effect in plasma. The more drug you take, the more it is in plasma. How about into the tumor? Does it actually cross into the tumor, across the blood-brain barrier? Yes, it does. And what you can see is that 20 milligrams, you have about 40% of the drug that's in plasma you find in the tumor. So it gets into the tumor, and the more drug you take, the more drug you find in the tumor. So great. So we know that the humans were like the mouse model. The drug does cross, gets into the tumor. The second question is, does it bind to that RTS promoter? Does it turn on transcription of interleukin-12? So this data shows you that it does. When you have no drug on board, when you're not taking the drug, there's no interleukin-12 in blood. Now, if you take 20 milligrams of the drug, all of a sudden you see this spike of interleukin-12 in the blood. It's about 25 picograms per milliliters. If you take more drug, 30 milligrams, you can see that the levels go even further up. If you take 40 milligrams, you're way up to 100 picograms per milliliter. If you stop the drug, the level of interleukin-12 drop again. Now, interleukin-12 makes a number of cells in the immune system generate interferon gamma. So when you treat T cells, macrophages, NK cells with interleukin-12, they start making interferon gamma, which is also a very powerful immunostimulant. So we also asked, can you see interferon gamma in the blood? And again, without any drug, you see no interferon gamma. Now, if you take 20 milligrams in blood, you start seeing that this interleukin-12 starts allowing interferon gamma to be made. And the more drug you take, the more interferon gamma in the blood. When you stop taking the treatment, levels fall back down. Now, these levels are really high in the blood. And that is why this became so toxic to patients at 30 and 40 milligrams. These are levels that you see very commonly in patients that have this cytokine release syndrome. And this really gives you the serological data of why we saw toxicity at the highest doses. So let's start looking at some survival. So the 20 milligrams was well tolerated. Let's look at survival of patients, again, recurrent GBMs. And we're comparing your survival of patients that took 20 milligrams versus those that took 10, 30, or 40 milligrams, the doses that really were not effective. And you can, when you look at this, you can see that there seems to be a clear-cut difference in survival. These patients seem to be doing much better with a median survival time of about 13 months or so compared to the ones that had the 10, 30, and 40 milligrams. So for recurrent GBM, some of these were second or third recurrences. 13 months is not bad. So after the fact, we went and tried to find out if there's anybody in these curves that does better. And one of the questions we asked is, if patients were on dexamethasone during the time they were taking the valilimex drug, was there a difference in survival? So it turns out that patients that were taking very low doses of dexamethasone, in other words, 20 milligrams or less during the 14 days of valilimex. So if you think about it, 20 milligrams divided 14 days, that's a dose of 1 to 2 milligrams per day. These patients did the best. Their median survival time was in the order of about a little bit over 18 months. Again, we're talking about recurrent GBMs, recurrent GBMs. But if you were taking more than 20 milligrams of dexamethasone, your survival was much less, and it was less than 8 months or so. So a big difference in survival. So again, dexamethasone seems to have a negative impact in this group of patients. Now, in some patients, we treated them with this interleukin-12, and lo and behold, they seem to progress. So here's one of my patients. We had taken out a recurrent GBM from the occipital lobe. This is post-op. We had done the injection already. And then we just followed this patient. He took this for 2 weeks of valilimex. And then that's it. That's all he's done doing. And now we follow him. And what we see is that in the parietal lobe, above the occipital lobe, there's this new area of enhancement. And then by week 8, this area of enhancement seems to be retrogressing a bit, but now there's a new area that starts appearing here, very close to the injection site. And at 24 weeks, this area is getting larger. So we took this patient back and we sected this as well as that, as this area of enhancement, these two separate areas of enhancement around the tumor cavity. And what we find is, yes, there's some tumor in these post-treatment tumor recurrent GBMs, but there's also a lot of inflammatory cells. There's a lot of immune cells. So this is one of these immunomics immunofluorescence stainings to look at different markers such as CD3, CD8, PD1, FOXP3, CD56. And here is just basically a comparison. So here we had 3 patients, a baseline before they got the interleukin-12 gene therapy, and after the interleukin-12 gene therapy when they seem to recur. And we're looking at, for example, how much CD3 plus T cells or CD3 plus lymphocytes are in the tumor. And you can see that there's many more after treatment. And a lot of these are CD8 plus positive. CD8 plus is a marker of activated T cells. So you see a lot more T cells infiltrating these tumors. Some of these are actually PD1 positive, again, showing that they are also exhausted. None of them are FOXP3 positive. FOXP3 is a marker of T regulatory cells, which are immunosuppressive cells, very few NK cells, and also a lot of PD-L1. So again, we see a lot of T cells infiltrating these tumors, but we also see a lot of evidence for what's called immune checkpoint signaling. The tumor is trying to shut off these T cells. What's also remarkable to me is that some of these patients seem to recur several months later, and when we go back and take out these tumors, there's evidence that there's lots of interferon gamma within these tumors. So again, a lot of evidence again that there's a lot of immunoactivation within the tumor itself. So the conclusion of this phase 1 trial is that you can use this viral vector to deliver interleukin-12. We see some encouraging biologic and immunologic activity in recurrent GBM patients. There's some encouraging survival in some patients, particularly in the ones in which very low doses of dexamethasone are being provided. After injection, the few patients which we took back show evidence of immune infiltrates, but there seems to be also evidence of evasion because of immune checkpoint signaling. So just like the other trial, we now started a new phase 1 trial where we decided to combine this with an immune checkpoint inhibitor. And so we've now evolved and gone from this to combining this with nivolumab. And this trial actually has finished accrual, so we accrued all patients to it. And now we're examining the data. So in this trial, patients actually got the immune checkpoint inhibitor, nivolumab, intravenously 7 days before the resection, by tumor resection. Just like the previous trial, they did get the drug validemax because we wanted to make sure that for some reason, the nivolumab did not stop validemax from getting to the tumor. And they continue with the validemax to turn on expression of interleukin-12, but they also continue with the immune checkpoint inhibitor every 2 weeks. So we're looking at the data of this trial, and this is actually going to be presented at ASCO in June, or at least virtual ASCO in June. I cannot give you, I already have the slides, I already know what the data is, but I cannot tell it to you too much because we're supposed to present it at ASCO. So there's going to be a virtual presentation, oral presentation of this at ASCO in the next few weeks. But basically, I can tell you that we treated 21 subjects. This is the abstract that's already come out. We treated 21 patients, and we did a dose escalation. So we escalated first one drug, and then a second drug, until showed that that was safe, and then we finally escalated the two together. We actually showed that this was very safe, so we can combine these two immunotherapies without much of an issue. All of the adverse reaction during this follow-up were consistent with immune checkpoint inhibition. They were all manageable. They were reversible. We can show, just like the other trial, that you can see interleukin-12 going up in the blood of these patients. We also could show that there was the combination of these two immunotherapies increased the amount of circulating T cells in the blood of these patients. We also see evidence, again, of pseudoprogression, where there are some patients where I seem to have increased inflammation within these tumors that then decreases with time. And so we're still looking at this phase 1 data, but now that we know that this is safe, we've now progressed to a phase 2 trial, multi-institutional, where we're combining this gene therapy with surgery in combination with an immune checkpoint inhibitor. And actually we changed from nivolumab to a different one. And this is a large phase 2 study that's involving multiple institutions. And we've almost finished accrual of this trial as well. And so we should have this data in the next year and a half or so. I see there are 3 questions. So maybe, no, never mind. Okay, so the last 5 minutes, I'm going to tell you about a different trial. And this is one in which instead of delivering a gene like interleukin-12 thymidine kinase, we're actually using a modified virus. And this is based on herpes simplex virus type 1. And the idea, just like I said before, is to have the virus infect lyse tumor cells, spread the infection throughout the tumor. But this process also allow for better T cell recognition of tumor antigens, so that you can get long-lasting vaccination immunity against the tumor. So this area, again, is showing some nice results. So there actually is an oncolytic virus based on herpes that's approved for melanoma. And this was the clinical trial that showed in metastatic melanoma that this oncolytic virus, which is called now Imloject, increased survival of patients. A few years ago, the group from Duke published the results with another oncolytic virus called poliovirus. And what they showed in this paper in New England Journal of Medicine was that patients that had convection-enhanced delivery of this poliovirus into their recurrent GBM, there was this tail of patients that seemed to survive for several years. And now they're in a large phase 2 trial of this treatment. Another oncolytic adenovirus called Dnatrix is completed a phase 2 trial in patients with recurrent GBM, and we're waiting for the data from this. I got this from the company, again, showing these tumors that have been injected with the oncolytic adenovirus, and then week 24, you can see that there's reduced enhancement of the tumor, reduced flare in T2 abnormalities. So some interesting data from patients. In Japan, my friend Tomoki Todo has this oncolytic herpes virus that I think just got approved by the Japanese FDA, and basically injection into the tumor of this oncolytic herpes virus called G47 delta causes it again this disappearance of gadolinium enhancing tumor over a period of about 2 years or so. So we have our own that we made, and I'm not going to go through a lot of details here, but this is one that was made in the lab by my group back in 2005. That's how long it took us to get this into clinical trials. It's based on herpes virus type 1. It infects and kills primarily glioblastoma stem cells. And we showed in animal models that this was fairly effective. So we wanted to show that when we inject patients with recurrent malignant glioma, that they did not come down with a neurovirulent toxicity by herpes, basically. So as of March, we actually finished this phase 1. We treated a total of 30 patients. The American FDA made me start at a very low dose of virus. This is 1 million viruses injected into these tumors. So in this trial, we did not remove the tumor. We just injected the tumor with the virus stereotactically. So we started at a dose of 1 million viruses. Some of these tumors were large. They even measure 8 centimeters. And we went up all the way up to 10 to the 10th viruses, which is 10 billion viruses. We really showed no evidence of toxicity or dose-limiting toxicities. There's one serious adverse event where 2 weeks after injection, we saw one patient developed a hematoma at the injection site that caused some seizures. But otherwise, this was very well tolerated. And so far now, we started the first patient in 2017. So far we have about 12 out of the 30 patients are still alive, and we're following them as we prepare for the next phase. These are the events. Most of them were not attributed to the virus, but things that happen in patients with tumors. And this is the only one that had that hematoma shown as possibly related to the virus. Just to show you, everybody shows you the best patient possible. So this is actually patient number 3. This is a gentleman that had a initial right frontal glioblastoma that we have resected. The tumor recurred right here, but he also had a second recurrence. So now he has 2 tumors, or 2 gadolinium-enhancing tumors. So we injected this new one with the virus. We used the MRI guidance in the intraoperative MRI suite so that we can see where the injection is. And then over a period of about 280 days, you can start seeing that both of these tumors, we only inject one, both of these tumors seem to start retrogressing, actually disappearing. This was actually called a complete response. This gentleman was not treated with anything else, was off steroids, went back to work full time. And unfortunately, about right after this scan, he recurred with a new tumor in the basal ganglia, in the right basal ganglia. We thought it was tumor. We biopsied it. Unfortunately, the biopsy, because it was in the basal ganglia, led to a small internal capsular stroke that left him with a left hemiparesis. He decided at that point that he did not want to have any more treatment and basically went into hospice and passed away 15 months after this. So this was a fairly dramatic, complete response from a patient with evidence of 2 GBMs. So this is really my last slide. I can show that we can inject these recurrent GBMs with these oncolytic herpes viruses up to very high doses. In some patients, we've taken them back. We can show that there's virus still present even several months later. we can show that there's lots of T cells in these glioblastomas. So again, we do turn them from cold tumors to hot tumors. And the survival data seems to be encouraging, but clearly we will need more advanced clinical trials to show that that is the case. So in conclusion, I want to thank people in my laboratory that have helped develop this virus. And these are all our collaborators over the years, developed us not only with the viral therapy, but also some of the gene therapies, particularly Advantagene with the first one, with GMCI, and then Zeopharm with interleukin-12. So thank you very much for your attention. Hopefully I didn't take too much of my time, and thank you.